Hi, I'm Dr. Bruce Emlich, and I'm here to present to you our paper on understanding the impact of the deep space environment on crop production. This paper is authored by myself, Deborah Wells, Ray Wheeler, Joy Amasa, Ye Zhang, Ralph Fritsch, Dinah Demopoulos, and Charles Quincy. We all work at the Kennedy Space Center in Florida, and this work is funded by the NASA Space Biology program. Really what this talk is about it's about a challenge that's facing NASA. As NASA seeks to put astronauts on the moon and Mars for long-duration missions, it's important that the astronauts are more self-reliant than they've ever been before. And it's a huge problem. Really, it's a food problem. And this is such a big problem that NASA is taking two different approaches to solving the problem. One is to a more open innovation approach, which will result in a centennial challenge that'll come out, I believe, in January of 2021, where teams will compete for a prize to develop systems, sustainable systems that help astronauts feed themselves. The other approach is the more science-based approach, which is really what this talk is about today. In order to understand the integrated long-term effects of the deep space environment on biological systems, we really need to look across a variety of platforms, and those platforms are really shown up across the top of this slide. And by looking across those platforms, then we can isolate the effects of things like gravity, radiation, magnetic fields, and this fourth factor really has to do with both the duration of the mission and then also with growing crops inside an enclosed environment. If we look, for example, or just a moment at the International Space Station, you can see that it orbits inside the Earth's magnetic field, so there's still a magnetic field present, but it has microgravity, which is a component that is difficult, if impossible, to gain on Earth. So really, it's only by looking across all of these different locations or platforms that we'll be able to puzzle out the impacts of each one of these factors. So what do we need to know in order to grow crops in space in these environments? What do we need to know in order to get to where we want to be? Well, really, to do that, we have to first identify our knowledge gaps. And it really helps a lot to have a diverse community to help identify those gaps, and also at the end to help fill some of those gaps. It would also help to understand a little bit about what those opportunities might look like for doing science in the future. Right now, NASA is assembling the diverse community, and this is what that community looks like. It starts off with space scientists, people who've done a lot of work on plants in space so far, plant microbiome scientists, people working on the space platforms, things like the Gateway and the human landing systems, science hardware designers, these are really the carriers for the science that will go into those vehicles, people who are experts on the space environments in those locations, and then also cutting-edge science tool developers. And the reason, or what I mean by that, is think about sequencing for a moment. It wasn't very long ago that it took millions of dollars and over a year to sequence a single human genome. Now we can do that same work in just a few hours and for just a few $100 in a very small format. And really, it's that small format and that low power need that will allow the cutting edge science to be done on the surface of the moon and beyond. So what do the opportunities look like? And I want to emphasize for a minute that this is really a very partial list of what the expected platforms and facilities and opportunities are going to look like. First, of course, we'll have the ground control chambers and the microgravity simulation facility located at Kennedy Space Center. We also plan to continue using the ISS at least through its entire lifetime. CubeSats are becoming ever more present in these endeavors. The CLIPS landers are supposed to hit the surface of the moon in 2022. Gateway will be in orbit of the moon, and it will be serviced by the Orion, used for the Artemis missions, which would be the first to return humans back to the surface of the moon. And to do that, of course, we need human landing systems. Where is it we want to go on the moon? NASA seeks to land near the Shackleton crater. And the reason for that is that it has these eternally sunlit regions around the edge of the crater, which are good for generating solar power. And of course, these eternally shadowed regions, which are believed to be rich in water ice. Currently, there are 3 human landing systems under consideration to get us down to the surface. Only one of these will be ultimately chosen. The first of these is Blue Origin's integrated launch vehicle, and this is led by Blue Origin and the national team, which is Lockheed Martin, Northrop Grumman, and Draper. The second vehicle, very different from the first one, is the Dynetics Human Landing System, and this is being built by 25 subcontractors who are all veterans in designing space hardware. And the third one is, of course, the Starship, which is being built and designed by SpaceX. These vehicles have very different sizes, shapes, and capabilities, and will provide very different opportunities for doing science. So what to expect? Well, to tell the truth, there are a lot of things we don't know yet. Simple questions like, what's the pressure inside the spacecraft going to be? It's really helpful if the pressure in the spacecraft is only about 1/3 of an atmosphere, because if you need to suit up to go out on the lunar surface, then you don't have to do any pre-breathing at those pressures. The temperatures and atmospheric compositions in those vehicles are going to be human-friendly when humans are present, but they may not be biology-friendly at all when humans are not present. And the actual mission profiles are still on the drafting boards at this stage. We've already talked a little bit about how unique each of the space environments are, particularly in terms of magnetic fields and gravity and radiation. And then, of course, anytime humans are involved, there's going to be some kind of life support system. And a key component of that is recycling the water, because water is very heavy and expensive to lift to these locations. Anytime you're recycling a lot of water and air, then trace contaminants build up. Now, these next several slides, I'm not going to go through in any great detail, but there are trace contaminants that have already been identified from spacecraft like the ISS. In addition, we know that gravity has a big impact on plants in space. And what this chart shows is if we look at low percentages of gravity, say 10 or 20%, we can already see that the boundary layer over the surface of the leaf is getting quite thick. And really that shows the importance of keeping an airflow rate of about 1 meter per second over the surface of that leaf so it doesn't suffocate in these low gravity environments. Also, we know that the CO2 levels in these vehicles is often very high, especially when compared to Earth. So at these super elevated CO2 levels, on this chart we have light shown in these for each of these different curves. So 400 micromoles, 1,000 micromoles, and 10,000 micromoles. And what's obvious is the stomatal conductance, or how much gas is coming through the surface of the leaf at a given time, is changing very much under those different light conditions. But importantly, it's not even going in the same direction. It's bouncing in different places, in different directions, and that's all an impact of these high CO2 levels. At low pressures, then we have another factor to consider, which is the diffusion coefficient. It becomes a lot higher at low pressures. And what that really means, in practical terms, is that water is going to be evaporating from the surface of the leaf at a much higher rate. So the transpiration coming off the plants is a lot higher, and that's going to have a big impact on the plant physiology. In addition, we will be growing the plants under LED lights, and the reason for that is LED lights are simply the most efficient way to generate light per unit energy at this time. LEDs, of course, have very specific color patterns, and we know that color is another important factor in how plants respond to their environments. So we're looking at the impact of light and light colors on plants, and we're working with outside companies in order to help us to understand these impacts and effects crop by crop. So speaking about crops, crop production in space has a whole range of challenges, and those are kind of listed here on this slide from Joya Massa. She's also indicated on this slide our current focus areas, which are fluid movement, radiation, high CO2, and the plant microbiome. We've already talked about several of those, but the plant microbiome, why is that important to us? Well, it's really important that the astronauts are going to be able to eat the food that they produce up there. So it's important that we have healthy microbiomes to make sure that that's possible. NASA doesn't really have a standard for food safety for things like crops because we haven't grown that many in space yet. They do have standards, however, for non-thermostabilized foods. This would be something like a candy bar or, say, M&Ms. And the orange limits and the blue limit lines there are basically for your aerobic plate counts and for your yeast and mold counts. And in the same way, the orange and blue bars, that's what they're showing across this chart, how many colony forming units you had. By and large, things were pretty good in this particular study, but there was one case where things exceeded the limits. What would be helpful is to have a microbiome present on those plants that helps to keep down and out human pathogens from those plants so that we know that they're safe to eat. The other reason microbiomes are important is they can really help with plant yield, and they can also help with the plants cope with stress while in space. What little work's been done in space on microbiomes, some of that's summarized in this slide. And again, I'm not going to go into this great detail, but what you can see is that the microbiomes for the flight are very different from the ground. And this is due to the completely different environments that we have, fully enclosed with astronauts floating around and then simulation chambers on the Earth. Other technologies are also going to be needed. We'll need to have good ways to plant the crops. And so here on the bottom in this picture is shown a polymetric seed tape that was being championed by Trent Smith at the Kennedy Space Center. It will help you handle small seeds and make it easier to plant or replant plant carriers in a space environment. Right now we're doing all we can to commercial growers and seed producers, and we're also trying to understand the best ways to sanitize the seeds and the different needs that different crops have. We can't ever forget why we're doing this work, and part of that is about having a happy crew and also a healthy crew. And what's shown in this image below is a little bit of lobster that's been applied to a surface of a lettuce leaf, and that lettuce leaf is really providing a crisp and nutritious component to a meal that they wouldn't otherwise have. Also, there's a psychological benefit to growing plants in space, and that's one of the reasons we really love Peggy Whitson, is because that Peggy really enjoyed gardening and growing cabbage for us on the International Space Station. So now it's really the time for the call to action. Please join our diverse community. Help us to identify these major gaps in our capabilities. Help us to outline the key science questions that need to be answered, and help us to understand what can be achieved on early high mass and power constrained missions, as well on later missions when more resources and more space might be available. If you're a member of the ASGSR, you would have already received an email from us asking you or inviting you to attend these optional 1-hour learning sessions. And these are going all through the month of October. Really what we're asking people to do is we're asking the scientists to attend the hardware and platform sessions and the engineers to attend the science sessions. And the reason for that is we want to get everybody on the same footing so that everybody can understand what the problems are that face the other groups. Only by doing that can we design the best hardware and the best science for these vehicles. Ultimately, this will result in some virtual workshops. The first of those will come right after the ASGSR meeting. It will focus on research campaigns. And the second will come probably in January of 2021, and it will focus on transformational research, the flagship and multifunctional hardware pieces. What all this means is white papers. White papers are necessary for the next decadal survey. Those papers will be due probably in the spring of 2021. So this is your chance to influence NASA. Join the community, submit white papers, enable the best science, enable the best hardware. And if you need to know more about how to get involved with this, please contact me, bruce.m.link@nasa.gov. Thank you for your time, and once again, this work has been funded by the NASA Space Biology Program. Thank you.